first thing that we will show you is the structure of DNA. This is the chemical structure of DNA. The building blocks of DNA are known as nucleotides. They consist of a backbone, which is a deoxyribose, and a phosphate group. It also consists of nitrogenous bases. There are 4 nitrogenous bases: thymine, adenine, cytosine, and guanine. There are also 2 groups of nitrogenous bases, pyridamines and purines. Cytosine and thymine each have a single 6-member ring and are considered to be pyridamines. Guanine and adenine each have a double ring made up of a 5-atom ring attached by 1 to a 6-atom ring. These are considered to be purines. One purine will match up with one pyridamine by a hydrogen bond on opposite strands of DNA. Specifically, guanine is the complementary base of cytosine and adenine is a complementary base of thymine. On our candy model, these bases will be represented by colored gumdrops. The hydrogen bonds between these bases will be represented with toothpicks. The phosphate group is represented by a blue Twizzler and the deoxyribose is represented by a red Twizzler. The DNA double helix is a double strand and runs in opposite directions. One end is called the 5' and the other is called the 3' end. The way in which we can determine which is the 5' end and which is the 3' end is by looking at the carbons. The 5' end will end with the phosphate group while the 3' end will end with the deoxyribose. The antiparallel structure of DNA refers to how the strands run in opposite directions. One strand runs from 5 to 3' and the other strand runs in the opposite direction from 3 to 5'. At the end of each double helix, one strand is 5', the other is 3'. The antiparallel structure of DNA is important in DNA replication because it replicates the leading strand in one way and the lagging strand the other way. One of the main ways DNA's antiparallel structure affects replication is in the way DNA polymerases build the new strands of DNA. The next stage we will show you is DNA replication. It occurs during the S phase of the cell cycle. The first enzyme involved in DNA replication is the helicase. The helicase unwinds and unzips the DNA in preparation for replication. Replication fork is the point where the DNA double helix is opened up. Single-strand proteins temporarily bind to each side to keep the strands separated. The leading strand, which runs from 3 to 5, is first replicated. DNA needs a primer, the RNA primase, which is something it can hook onto for building a new DNA strand. DNA polymerase III goes down the strand adding nucleotides, pairing them up with the existing strand, A to T, C to G. DNA polymerase I goes back and replaces the RNA primers. The lagging strand runs from 5 to 3. RNA primase once again gives DNA polymerase III a starting point to work backwards from. DNA polymerase III then copies a series of newly synthesized DNA fragments known as Okazaki fragments. DNA polymerase I once again goes over the RNA primers to create unified DNA fragments. Then finally, DNA ligase seals the fragments into one long continuous strand. Since the DNA polymerase can only add bases in a 5' to 3' direction, the leading strand can continuously replicate. However, the lagging strand can only be replicated in fragments, therefore the DNA strand elongates in a 3' direction.
The 3 main differences between DNA and RNA is that DNA is double-stranded while RNA is single-stranded. In DNA, the backbone consists of deoxyribose and a phosphate group, while in RNA, the deoxyribose is replaced with ribose. In DNA, thymine is not a nitrogenous base. Instead, it is replaced with uracil, and as a result, adenine will have a hydrogen bond with uracil instead of thymine. In most eukaryotic genes, coding regions, or exons, are interrupted by non-coding regions known as introns. During transcription, the entire gene is copied into a pre-mRNA, which includes exons and introns. During the process of RNA splicing, introns are removed and exons are joined to form a continuous coding sequence. This mature mRNA is ready for translation. After intron splicing occurs, a poly A tail and a cap is added to the ends of the mRNA. Synthesis is transcription. During transcription, mRNA is created. Transcription must occur in the nucleus, where the DNA in the cell is located. First, the DNA must be unzipped and unwinded with the help of RNA polymerase. In our model, the orange Twizzler is the nitrogenous base, while the purple Twizzler is the ribose, which is the sugar in mRNA. It is analogous to deoxyribose in DNA. RNA will sit on top of the template strand, and DNA strands will reconnect after the RNA moves into the cytoplasm. The new RNA strand will match the coding strand, except wherever there's a T, there will be a U. It uses DNA as a template to make an RNA molecule. RNA then leaves the nucleus and goes to a ribosome in the cytoplasm, where translation occurs. Translation reads the genetic code in mRNA and makes a protein. Translation occurs in 3 phases. The first phase is called initiation, where the ribosome assembles around the target mRNA. In translation, the mRNA slides between the 2 subunits of the rRNA. The tRNA must match up with the mRNA's codons. After matching up, the amino acid from the tRNA will transfer onto the polypeptide in the P site. This process will repeat multiple times. Through elongation, amino acids are added to the polypeptide chain, elongating the polypeptide. Later, this polypeptide will be released as a protein into the cell. The polypeptide is a chain of amino acids represented by fruit snacks. These amino acids are connected by a polypeptide bond. When 2 amino acids join together, the carboxyl group and amine group creates a dipeptide plus water. There are 3 types of RNA: mRNA, tRNA, and rRNA involved in the process of translation. tRNA, also known as transfer RNA, will carry amino acids. It has an amino acid at one end and a sequence of 3 nitrogenous bases or codons at the other end. rRNA, also known as ribosomal RNA, is made up of a small subunit and a large subunit. On the large subunit, there are 3 sites, the E, P, and A site. The E site stands for the exit site, which is where the tRNA leaves the ribosome. The P site is the polypeptidal site, where the amino acids form into a polypeptide. The A site is where the tRNA binds to the mRNA and is holding the next amino acid that will be binded onto the polypeptide chain. mRNA, also known as messenger RNA, is single-stranded and has alternating ribose and phosphate groups as its backbone, and also 1 nitrogenous base bonded with a ribose. Anticodons are the codons that are attached to the tRNA. If they match up with the codons on the mRNA, then the amino acid from the tRNA will be added to the polypeptide. There's a universal genetic code that can be used when deciphering the amino acid. After the tRNA and mRNA match up, the 3 nucleotides from the mRNA will form a certain amino acid depending on the sequence.
With an inversion, part of the sequence of DNA within the chromosome becomes oriented in the reverse direction with no molecular loss. An inversion is not a problem if it does not disrupt a crucial gene region, but it mispairs during meiosis, so it can lead to chromosome deletions and gametes.
A substitution is a mutation that exchanges one base for another. The exchanging of bases have varied results. For example, a substitution that changes a codon to one that encodes a different amino acid is called a missense mutation. This results in a small change in the protein produced. If the properties of the amino acid stayed the same, then it is considered conservative mutation. And if the properties are altered in the amino acid, then the missense mutation will be a non-conservative mutation. On the other hand, a substitution could change a codon to one that encodes the same amino acid, causing no change in the protein that is produced. This is referred to as a silent mutation. Also, a substitution could change an amino acid codon to a single-stop codon, causing an incomplete protein. The protein will probably not be able to function. This is known as a nonsense mutation. Insertion mutations and deletion mutations add or remove one or more DNA bases. Insertions and deletions can shift the reading frame of a gene, changing the grouping of bases into codons. These changes can greatly affect a protein's amino acid sequence. Sometimes an insertion or deletion could result in an early stop codon, so a frame shift typically causes the wrong amino acid sequence, but occasionally causes an incomplete protein. Insertion and deletion are all part of frameshift mutations. During insertion, one nucleotide, thymine, the green gumdrop, is being added in the middle of the template strand. As a result, through the process of transcription, thymine's base pair, adenine, the red gumdrop, is added to the mRNA at the same location. Therefore, during translation, the amino acid sequence is altered with this new strand of mRNA. At first, the mRNA would have produced proline, but after the mutation, the codons form threonine. In deletion, the purple gumdrop, guanine, was taken out of the template strand, causing its matching nucleotide, the white gumdrop, cytosine, to be taken out of the mRNA. Before this occurred, the mRNA would produce proline. However, after mutation, as a result of taking out a codon, the mRNA produces leucine.